Welcome back to Bioinformatics Introduction and Methods from Peking University. I'm Liping Wei from the Center for Bioinformatics at Peking University. Let's continue with the introduction and history of bioinformatics. In Unit 4, I will give you an overview of this course. There will be 6 weeks in this MOOC, with 2 topics per week. I hope you still remember the biological, informational, and historical conceptual frameworks of bioinformatics that I talked about in Units 1 and 2. In Unit 4, I will try to put the content of this MOOC into the conceptual frameworks, which hopefully can help you understand the content better. I will try to tell you what are the biological questions addressed in each topic, how the biological questions were formulated into a computational problem, and when was the main methods developed. We will talk about sequence alignment. The questions are, how similar are 2 DNA or protein sequences? Could they be homologous and have descended from the same ancestral gene? Can we predict a newly discovered protein's function based on the known function of another protein? This question is formulated into how to find the optimal alignment or near optimal alignment between 2 sequences of letters, how to do it fast, how to quantify the similarity. As soon as 2 or more proteins were sequenced in the 1960s, sequence alignment has been a central problem in bioinformatics research. To solve this problem, the classic Needleman-Wunsch global alignment and Smith-Waterman local alignment algorithms were published in 1970 and 1981, respectively. The PAM matrices that quantify the goodness of fit of amino acid substitutions were published in 1978. Later, another set of substitution matrices, the Blossom matrices, were published in 1992. Dr. Michael Waterman will tell us more about the historical context of the Smith-Waterman algorithm. We will talk about sequence database search. Among all known genes, which was the most likely homolog of my gene of interest? Can I use what's been discovered about other known genes to guide the functional study of my gene of interest? The challenge is, from tens of millions of protein sequences or trillions of nucleotide sequences, how can I find that 1 or a dozen or so protein genes that are similar to my gene of interest? It is like looking for a needle in a haystack while the haystack is growing exponentially. How can we solve this technical challenge? In 1970s and 1980s, the sizes of the protein and nucleotide sequence databases gradually exceeded the analysis capacity of the existing algorithms. Fortunately, the BLAST algorithm published in 1990 and the later gapped BLAST and side-BLAST algorithm published in 1997 provided elegant solutions to these problems. How should we store a huge amount of data? We will talk about the concept of ontology. After the data is stored, how can we use the data and statistical tools to find statistically significant patterns? We will use the identification of molecular pathways as an example. If an experiment found 300 or 400 genes that were statistically significantly differentially expressed between tumor and normal tissues, which protein families and which metabolic and signaling pathways were most relevant? We'll talk about the gene ontology, the tech database, and the COVID software that we developed. After learning about all these algorithms and software, how can we apply them to study real questions in life science? One of the power of bioinformatics is that it can make predictions based on existing knowledge. One of the methods to make prediction is based on Markov model and hidden Markov model. We will introduce what are Markov models and hidden Markov models, how to build these models, how to train the models, and how to predict with these models. We will show you how the Markov model can be applied to sequence alignment, how to find the coding region given a DNA sequence using hidden Markov model. We will discuss how to extend the basic models we introduced here to do gene prediction in practice and how the widely used gene prediction tool GenScan works. There is a wide variety of bioinformatics resources. What are they and where can you find them? We will give you an overview of important databases and software resources in bioinformatics. We'll focus on the large, active, and popular resources. We hope that the overview can be a useful starting point for your research. To sequence one person's genome, next-generation sequencing technologies generate about 900 million 100 base pair short reads. How can we reconstruct this person's genome and identify his or her genetic variants using the reference genome as a template? The per-base error rate of these reads may be 10 to 100 times as high as that of traditional Sanger sequencing. How can we align the 900 million error-prone reads to the reference genome and distinguish the true genetic variants from sequencing errors? Methods such as MAQ, BWA, and GATK, published around the year 2000, provided solutions to these problems. Dr. Maynard Olson from University of Washington will share with us his personal experience of bioinformatics in the early days of the Human Genome Project. Once we identify a person's genetic variations, how can we predict whether they will change the protein structures or function? Will they cause diseases? This question can be formulated as a supervised classification algorithm in machine learning using existing knowledge as training data. We will talk about the SIFT and polyphon algorithms as well as separate, an algorithm developed in our own lab. Another application of next-generation sequencing technologies is to profile gene expression at genomic scale. We will cover how to use next-generation sequencing to detect which genes are expressed, which genes are differentially expressed between different tissues or between tumor and normal tissues, what splicing isoforms exist. How can we calculate gene expression level from the short sequencing reads? How can we calculate the statistical significance of the difference between the gene expression levels of two genes? How can we assemble the short reads into isoforms? Cufflinks and the method by Drs. Gao and Lei Kong, SPEP, were developed around 2010 to solve these problems. A surprising discovery in the past 10 years from the expression analysis was that there are numerous expressed and sometimes even spliced transcripts that do not encode proteins. Instead, they exist as non-coding RNAs. We will talk about how to determine whether a transcript code for protein or not, and what are the likely functions of the non-coding RNAs. How we can use knowledge about known coding and non-coding RNAs to build predictive models of coding potential. How can we find the features of the non-coding RNAs? We will discuss the popular CPC algorithm that we developed in 2007 and other related works. How can you study a biomedical question using a bioinformatic approach? We will demonstrate this using two case studies, origination and evolution of new genes and the evolution function analysis of DNA methyltransferase. We will show you how to integrate multiple sources of data and software to find interesting patterns and functional elements. We'll focus on common software and data and common problems. These case studies came from work Dr. Gao and I had done within the past few years in collaboration with Dr. Manyu Long from University of Chicago and Dr. Gang Pei, president of Tongji University. They had both kindly come to this MOOC and shared their work and perspectives with us. I hope that you will enjoy this MOOC. Happy learning!